good to go? Okay, cool. Then we can go ahead and get started. All right, so hello everyone. Hi, and hello people that are seeing us through the camera. Good afternoon. My name is Dr. Rita Blake. I am the Manager of Education at the California Nanosystems Institute here at UCLA. It's the building right next to this one that you guys all probably parked next to or near. Welcome to the final presentations for Nanoscience Lab Session A of 2017. Just to give you guys a little bit of primer before we start, I wanted to tell you a little bit about CNSI, about the people behind the scenes and in front of the scenes working to make all of this work possible, and then also a little bit of background as to what your students have been doing for the last 5 days, because they have had a busy schedule. So, and I'm sure they'll attest as to how hard they worked. So normally I would say welcome to CNSI, but since we're in La Kretz, welcome to La Kretz. But it's right next to CNSI where these students were pretty much all week. It's the California Nanosystems Institute. So it's a pretty interesting place on campus, actually. It's not a department like most of the buildings that house departments here at UCLA. It's actually an interdisciplinary research institute that's focused about the intersections between the different disciplines with a focus and a concentration on nanoscience. So it's a facility that brings in researchers from all of the buildings that surround it, from physical sciences, from life sciences, from School of Engineering, from the School of Medicine, from the School of Math, to come together and look at this field, this very burgeoning, interesting field called nanoscience. So back in the early 2000s, when we had a governor by the name of Gray Davis, and when California still had a lot of money, he decided to open what were called California Institutes of Science and Innovation that were dedicated to making these research institutes that were looking at the next big thing in science. So CNSI is one of them. So we have one here and one in Santa Barbara that, again, are focused on looking at nanoscience. So there's a lot of cool stuff that happens at CNSI. We've got a lot of faculty that come together to do interdisciplinary research projects around nanoscience. We've got downstairs in the basement, we have a lot of core laboratory facilities that are dedicated to the making and the studying of nanomaterials. And our students this week got to see a few of those spaces and get some hands-on insight into what happens down there. And also on the 6th floor, we have an incubator space. So we actually also foster innovation, not just in a traditional academic type of setting, but also in an entrepreneurial setting. So researchers who come up with ideas that have some real commercial potential can utilize those ideas and develop them and bring them to market using our incubator space as an area to take the idea from something more nascent to something that's much more scalable and producible. So the 4 sort of main things that CNSI does, one is team science, again, this sort of interdisciplinary research that happens between faculty. We have the core technology centers, the core labs, again, dedicated to the making and the looking at nanoscience. We've got the entrepreneurship, and we also have a big focus on education, which is my area. So we do a lot of work creating nanoscience workshops and experiments that we teach to educators that then go on and teach it to their students in the classroom. We do a lot of work on public outreach, everywhere from schools to malls to all sorts of stuff in between, to science centers. And we do a lot of work in trying to develop new modes for students outside of the classroom through entrepreneurship competitions and the like, to really get people to understand this sort of multifaceted world of science and nanoscience. So there are quite a few people who are with CNSI that really made this possible. First and foremost is our faculty director of the outreach program, Dr. Sarah Tolbert, who I will invite to come up and say a few words if she will be so willing. So please give Dr. Tolbert a nice welcome.
I am shorter than Rita. So it's a pleasure to be here and get to welcome you to our final presentations. As the director of this outreach program, I get to be involved with a lot of different aspects of our program. So as Rita mentioned, we work with high school, middle school, and elementary school teachers from around the greater LA area. Basically, anybody who'll get in a car and drive here can be a part of our program. And we also do a lot of public outreach events where we just try to expose the broader population to what is so cool about nanoscience. But I think my favorite part of our program really is our summer programs, because this is the time when we get to bring people into our home, into UCLA, to really get a deep dive into what is nanoscience. And we also get to take advantage of some of the cool facilities that UCLA has. And when we go out to a shopping mall, we can bring a few samples, but we can't bring a $3 million electron microscope and show people how to actually see atoms and see nanoscale structures. And so you guys didn't really look at atoms, but you did look at insects and other disgusting things at very high resolution. And I think that the ability to both do hands-on experiments and get to play with some of the really great toys that we have here at UCLA, and scientists call our equipment toys in the truest sense of appreciation, that this is what we get to do, is use this really great equipment to both play and learn at science. And so it's a real pleasure to have you guys be able to come here and be a part of our program, and also to get to interact with all of our really phenomenal graduate student mentors who've been teaching you across the course of this week. So I hope that you've gotten a chance to really chat with them about what it is like to be a scientist and be a graduate student, and I hope it has given you some sense of what you want to do with your career path in terms of going on in STEM-related fields. Maybe you fell in love with it. That'd be great. Maybe you at least learned you hated it, which is knowledge in itself and tells you something about what you want to do. But hopefully you got to see really the diversity of fields that are available to you from a range of fields, not just the traditional chemistry, physics, and engineering, but all of these interdisciplinary science. And so our goal for today is that you're going to see something about what they learned about interdisciplinary science. Rita is going to talk more about their actual homework assignments and what they did, but they're related to trying to make science accessible to the general public and visualizing science. And often when we start with this homework, people are like, this isn't like real homework. This is like not what scientists do. But in fact, this is what scientists do, and it's what scientists need to do more of. And there's a wave of scientists going into politics right now because they have realized, oh my goodness, the world does not understand and appreciate what science has to offer to our society, and we need to change that. And so what I ask of you guys and you guys is, independent of political leaning, bring science into the discussion, because I think it's a really important part of what we can do as a society. And so now that you know how to communicate aspects of science to the general public, go start explaining things to everybody who will listen. This is my son right here. And unfortunately, since he was like 3, he's had to learn how everything on the planet works. And it's good. What I can say is you make very curious people if you start explaining everything. So if you have siblings, start teaching them all of the cool things about how the world works. If your parents are not scientists, start with them. They're a great audience. And so today we're going to get the beginnings of that in terms of how to make some of these cool nanoscience concepts accessible to the general population. Thank you.
So before we get started, there's a few other people that I need to give some quick thanks to. Our CNSI director, Dr. Jeff Miller, and our executive director, Sonia Luna, who both provide immeasurable support, particularly administratively. We can't get this program executed as smoothly as we do without them. And also Dr. Jiaming Chen, who is the previous director, actually, of our education and outreach program, and now works at the Parker Institute at UCLA and has been kind enough to help me throughout the week with making sure that things run smoothly and lending his invaluable expertise. So, Ja, thank you so much for all of your help as well. Dr. Elaine Marita, I also need to give a huge thanks to. She's our program coordinator. You all probably got very helpful emails from her at some point, and she has also helped keep my brain straight and all of our instructors' brains straight as we try to make everything come together for this very, very packed 5 days of very intense work. So Elaine, thank you so much also for all of your work. And we had too many instructors and counselors for me to find photos of everyone to put them all in, but they are also truly the lifeblood of this summer program. One thing that I'll add to what Sarah was talking about is that all of the instructors, the graduate student instructors that you guys are working with over the last few days, they are the people that actually develop these experiments. So under the direction of Dr. Tolbert, they are the ones who actually are doing, real nanoscience in their labs, get inspired by what they do. They come up with some really cool idea that they think could make an interesting experiment. And then they work really, really hard over the course of a year and then even beyond to make those experiments go from idea to execution. And it is no small feat to make these experiments. So it's many, many hours of work prior to this program and then all the hours of work during this program where both the counselors and instructors lend their time and their expertise and their knowledge to all of you guys. So let's give a big thank you to all of our instructors and counselors. And also a thank you to all of you, to all the students for all of your hard work over the last 5 days, and to all of our parents and friends and family who are here to show up and support for the final presentations. Thank you guys as well. So you guys probably all got this at some point before you got here, but just as a reminder for how much work these students have been putting in, they've had a very, very packed schedule over the last few days. Every morning was a different nanoscience experiment that touched upon a different concept in a different field that nanoscience is very integral to and is very important for enhancing. So every morning they would do a new experiment, and then in the afternoon they would get a guest lecturer by one of our other graduate student volunteers or postdoctoral researcher volunteers, and then would get their hands dirty with some more sort of fun activities in the afternoon that were there to enhance what they were learning. So seeing the labs at CNSI, getting to do some extra things like spectroscopy or microscopy to be able to really look at the things that they were doing and they were making. Or doing some worksheets to just, really hone in on that understanding of the material that is very, very sort of unique and complex, and certainly stuff that they probably haven't encountered much in their normal school. And then every night they had a homework where they were, again, like what Dr. Tolbert was saying, taking those ideas that they learned, these very new concepts, and then trying to translate them visually into an explanation. So their task was to take all of this rigorous science and then create a representation of it, a metaphor, if you will, where somebody who wasn't familiar with the science could look at it and get some type of an understanding of what was going on. So that's the homework that they had to do every night. And today, what they're going to be doing in their presentations is each team of 3 has gotten a representation of somebody else's stuff. So they had a representation that was not of their making, and they had to look at it, figure out what that visual meant, and we'll try to explain to all of you guys what the visual is representing, what's the science behind it, why is it important. So, and they had basically an hour to get ready to do this. So it is a tough challenge, and they've been doing this all week, but in 1 hour it's not an easy task. So we will, I'm sure, all be very, very impressed at what they've been able to come up with. So just as a reminder for students and for everyone who's watching to be aware, so every group is going to have about 5 minutes to present their image, and then 5 minutes for some Q&A, and then I'll ask the original artist of the drawing to also stand up and say a few words about what the drawing was, and if there were any concepts actually that was not caught by the presenters, just to get some extra perspective, and to also acknowledge those people that created those drawings. Please, just as a note for you guys and to help the people watching at our live stream, please talk into the mic. We have 3 mics here. These 2 you can pick up and walk around, so you've got some mobility. Please, please, please talk into the mic. Otherwise, your parents who are watching afar, or your friends, or whoever else, will not be able to hear anything that you're saying. So please, please talk into the mic. And Sarah has something to add.
So I don't have a mic, but I'll have the details. We will also
Yes, thank you. So the Q&A is for everyone. The Q&A is for our students to ask questions, for our audience here to ask questions, for instructors to ask questions, for anyone who is in this room can ask a question. So we encourage everyone to be a part of the Q&A.
And just for those of you who want to help your dear children, if you don't ask questions, I'm asking questions.
So there will be questions asked. Someone will ask questions. Yeah, the less Dr. Tolbert will. All right, so with that said, I'm going to pull up the first representation. Oh. All right. So team one, please come up. And let's give team one a nice hand. And once again, just remember to talk into the mic.
Okay, hi, my name is Minji.
My name is Catherine. Sorry.
My name's Julia.
And we're going to talk about a topic that we covered on Wednesday, which was on biotoxicity. So for this lab, we were using silver and yeast particles to see the effect of silver on the yeast cell. And to do this, so we used 3 particles to do this. And the first one was silver powder, and the second one was silver ions, and the third one was silver nanoparticles, which were most effective.
Okay, so for our drawing, there are 3 main ideas. And so for the boulder, that represents the silver powder, and the wall represents the yeast cell. And so what happens is that the silver particles are trying to infiltrate the wall, right, which represents the cell. So since the boulder is too big, it won't go through. And the 2 smiling people on the left side over there, those represent the organelles, and they're happy because they're able to defend the yeast cell and prevent the boulder from infiltrating. And the 7 bars on the top left, that represents the health of the cell. And so since there's a full 7, that shows that the cell is pretty healthy and there's no damage done. And the middle section, that represents the silver cations. And since they're ions, they have to pass through the ion channel. And since they have to go through an extra step, the person is drawn shooting them from a slingshot. And these ions are able to go through, but only some of them are effective, which shows that some are past the wall and some aren't. And the health of the cell is decreasing, so it shows that the cell is getting damaged. And the organelles are a bit freaking out because there's damage done. And the last section shows the nanoparticles. And the giant one shows how there's a bunch of nanoparticles making up the ball. And these kind of show how, these nanoparticles kind of show how they're passing through the wall and they're able to make it through, and since they're disguised as the glucose molecules. And since they're able to infiltrate the wall, they splatter on contact with the cell membrane, which makes the nanoparticles destroy the cell from within. And so this effect is displayed by the 2 organelles being with X's in their eyes, which shows that they're dead. And yeah, so it's a bit morbid, but that's what happens. And then,
Okay.
yeah, and then also the health of the cell is decreasing, which is represented by the 2 bars. Yeah.
Okay, so basically how the silver is able to have this effect on the cell is because silver is used to prevent and protect against like bacterial growth and stuff like that. And the process of how they kind of do that is like basically all these different types of structures, like the powder, the ions, and the nanoparticles. So they all eventually are basically they're basically like they break down into like kind of how the ions are. And because these ions have a very high amount of like surface area, they're able to do the most damage. And so like the boulder in the example, so that represents the silver powder, and that will eventually break down into those silver ions. So it's eventually able to do damage, but but as it's when it's only powder, it's not able to really be effective. So, and then the silver ions, so those are able to do damage by just like mimicking kind of like the cations that enter the ion channels in the cell's membrane. And they're able to do some damage. But the nanoparticles, so what they do is when they make contact with the cell wall, they're able to release the smaller like silver ions, which are able to do damage to that, because the silver ions, so they catalyze the formation of reactive oxygen species. And what the reactive oxygen species do is they are able to react with the lipids, causing the membrane to break. So that's how the nanoparticles are able to infiltrate the wall. And so additionally, the display doesn't necessarily show exactly how the particles are able to actually like get through to the cell wall, because when you're talking about cells, like for example, like the nanoparticle. So the silver nanoparticles, because they're like a very similar shape to how glucose molecules are, the cell actually takes them in through endocytosis. So in a sense, it's a lot more effective than these other ways that are shown, which I don't think was exactly like explained as well through the drawing. And also, in addition to that too, we learned more about the surface plasmon resonance, which is basically like the when the molecules are smaller, the electrons, they move faster, which makes a higher frequency for the wavelength of light. And when they're bigger, the electrons make like a slower frequency, which means that the light will have like a lower frequency of like the wavelength will have a lower frequency. So that means that like the bigger the particle, like it's more close to red light versus a smaller particle, which will emit like a color closer to blue or violet. So I think we're open for questions now.
Are there any questions?
Yes.
So the silver is trying to go through the glucose or yeast cell because they're trying to destroy the yeast cell from within. So even though they go in through the membrane, they still kind of destroy the organelle and basically kill the yeast cell overall.
Yeah, they still have a toxic effect on what's inside the cell.
The middle section right there.
Oh, the bold, right.
Out of the way.
Yeah.
Okay, so you guys said, so in agreement with what I said on the first day when we talked about cartoons, they can never capture everything about an experiment. And so you described some things that were not captured, in particular the idea of the nanoparticles being eaten. So how could you possibly imagine modifying this cartoon to add some aspect of the experiment that was not already shown?
Maybe there could be like a gate or something that passes through, like to get like supplies or something like that, and maybe that could be like a way for like For example, like for like the ion channels, we could do something like that. So it's kind of more obvious that it's through like, it goes through something in order to get through to the cell, not just breaking through necessarily.
And in addition to that, maybe this person could have added an additional wall or something that represents the cell and the glucose entering in to show a comparison between the nanoparticles and the glucose.
And also, like, since the nanoparticles are kind of disguising themselves as glucose molecules, it seems kind of like the nanoparticles are just like a rock. It doesn't really act like the wall itself.
Um, well, like I guess the special thing about like the size of the nanoparticles that we're using is that they're like the same size as the glucose is. So it's actually, I believe it's more effective in that sense because it's kind of, it takes on a similar like shape and size to what like the cell wants, like the glucose that it wants. So.
Yeah, it's a cluster of nanoparticles.
So we actually heated a solution of silver ions. And as we heat the silver ions, they cluster into small particles. And the instructors told us that if we get like a slight yellow color, that indicates that we have small nanoparticles. Like Julia said before about this plasma effect.
Yeah, the plasma effect, the surface plasma.
Yes.
Well, so because like the size of the particles like determines kind of like the wavelength frequency, so we could like, are you asking why as in why yellow would be a good color or as in just Oh,
Because as the color gets darker, that means the nanoparticles are getting bigger. So since it's yellow, it means that the nanoparticles are still relatively small.
yeah, it means that too.
What's the actual size scale relative to each other, like from your largest molecule down to your nanoparticles?
So the silver powder would be the biggest, and then it will be the nanoparticles, then it will be the ions because the nanoparticles are actually composed of tiny silver ions. The nanoparticle would be around 10 nanometers, which is similar in size to the glucose. We're not sure
The
about
other
the other two.
So will the artist behind this drawing quickly stand up and add any insight that they would like to share about their drawing? So who is the artist for this one?
So the wall, I was trying to portray lipid peroxidation because it's slowly degrading, but the silver nanoparticles are able to get through the wall.
Thank you. All right. All right, we've got a laser pointer now. Yes, awesome. All right, so next group 2. And you've been doing great so far, but I'll just remind everyone again to talk into the mic.
Hi, I'm Millie.
Hi, I'm Nafkiran.
Hi, I'm Juliet.
So today we're going to be presenting the concept of hydrophobicity and hydrophilicity. So essentially a surface that is hydrophobic repels water and does not want to be wet. And in a hydrophilic surface is water loving. It does not repel water. And so we're going to talk to you guys about the nanoscience behind this concept and how we can measure hydrophobicity and hydrophilicity.
Okay, let's get into the math of this. Okay, so the way you measure if the relationship between a water droplet and it being on a surface is hydrophobic or hydrophilic is by measuring the contact angle. And so if you want to see if that surface on which the water is on is hydrophobic is by seeing if the angle between the water droplet and the surface is over 50 degrees. If it was hydrophilic, meaning that it likes water, as Millie said, the contact angle would measure to be over 15 degrees. Now, if you look at the super side, the extremes of both, a super hydrophobic surface would be over 150 degrees. And this would be a high contact angle because if you look at the surface where the water droplet is, the water is beaded up. And if you look at the super hydrophilic surface, the water will be spread out because it is a low contact angle, and that would measure out to be 15 degrees. And I'll give it over to Juliet.
If you guys want to use this, by the way.
We have a laser pointer now. So in this cartoon, there are, 4 panels. And each of these panels represent one of the 4 conditions that both Millie and Kieran spoke about. So in this first panel, we have a person. And in all of these panels, the people represent the water droplets. So in the upper left-hand corner, this person is laying down completely. And we even have a nice little contact angle measure there of 10 degrees, which would show that the angle is less than 15 degrees, and therefore the surface is super hydrophilic. And then in the next panel here, the person is sort of sitting down, not really standing up, but they're definitely spread out, and the contact angle measure is 70 degrees, and which again would be a hydrophilic contact angle measure, and so this surface would just be hydrophilic. And in this panel, the person is kind of standing up, but not completely on their tiptoes like in this next one, and the contact angle measure is 110 degrees, making this just hydrophobic. And then in the last panel right here, the person is completely standing on their tippy toes. They really do not want to be touching the surface at all, just like the water that completely beads up. And the contact angle measure is 160 degrees, which is greater than 150 degrees, making the surface super hydrophobic. Now, so far, we haven't really spoken about the nanoscience part of all of this. So you're probably wondering, what does it have to do with anything? So right here, we have all these little dots, and these dots represent the nanoscale roughness that would make a surface super hydrophobic, because when a surface has nanoscale roughness, it can either be super hydrophobic or super hydrophilic, and in this cartoon, we see that rough layer. And anyway, so when there's a nanoscale roughness, there are bumps, basically, and since there's air between those bumps, the air pushes the water up, and that's what causes it to beat up, and that can go either way. So it can go either way, and that's what can make a surface super hydrophobic as opposed to just hydrophobic. So all these different bumps here sort of represent the different levels of how it could be bumpy in on the nanoscale and either repel water or in this case allow the water to seep in because the bumps are right under there. So it's allowing the water to spread out on the surface.
Any questions? Dad.
Sent me the question, didn't you? This makes me think of like rain gear or camping equipment.
Mm-hmm.
How do you apply? What are some other ways to apply this, or am I even in the right ballpark? How do you?
You are in the right ballpark. They actually use nanoscience to create like rain jackets that specifically repel water. Like they create almost like a nano rough surface under certain rain materials. But also, for example, like often you see like a little droplet of rain on a leaf. And that is basically like a hydrophobic surface in most cases because the leaf is trying to concentrate that water in one specific location so it can then channel it to its roots. So that's a way in nature like you see this concept appear.
Can you repeat the question into the microphone for the internet audience because they can't hear?
Oh yeah, his question was whether or not, well, he was reminded of like rain gear and raincoats and was wondering whether or not this concept applies to that. Yeah. And to other stuff. Okay. Go ahead.
To add on to Millie's point, that leaf is the lotus leaf. We also experimented with this in the lab. And the reason why this happens is because a lotus grows in a river and that water is dirty. So it naturally wants to repel that dirty water off of its surface.
So it's hydrophobic. We actually used a protractor. Yeah.
Yeah, it was a really great experience the first day. Take out your cell phones and take pictures, please.
Yeah.
Yeah, so I have a bunch of pictures on my phone of these water droplets that we measured, and it was good. Yeah, that's how we measured it, just a protractor and a picture.
Well, we actually measured different droplets on different materials because some of the materials were more hydrophobic than others. So we were testing to see how this came out. It was cool. Yeah.
So you guys commented that nano rough things can be either super hydrophobic or super hydrophilic.
Mm-hmm.
So can you tell me how you might modify this cartoon to capture that essence?
And can you repeat the question for the audience?
Sure. Okay. So she mentioned how we had said that either a surface can be super hydrophobic. No, how a super hydrophilic surface can actually help a nano rough surface can make something either super hydrophilic or super hydrophobic, and how we can possibly modify the cartoon to represent that. And actually, we had an idea, Juliet and I did.
Yeah, so you can clearly see in you can clearly see in this panel that there is some nanoscale roughness, and that's definitely that's contributing to the superhydrophobicity of that panel. But we didn't really see that for the superhydrophilicity, so we thought that it would be more clear that this can apply to either condition if there was some nanoscale roughness for in the top panel where the surface is superhydrophilic.
Well, the first has a different angle than the other. Or, I'm not so sure. Maybe you could make the roughness look different. Maybe there could be more roughness on the one that's more hydrophilic.
So your question was, what's the difference between a super hydrophilic and super hydrophobic substance? Okay, I talked about this already. I believe so. So if you look at a super hydrophobic surface, the water will beat up, while on a super hydrophilic, yeah. That's the what? Right. Why? Because on a super hydrophobic substance surface, it doesn't like water. It wants to repel it.
Can I just add to that? So I didn't explain this clearly enough before. When there's a nanoscale roughness, there's air in between the bumps, and that air can push the water up, making the surface super hydrophobic. And then in the case of super hydrophilic, that space allows the water to spread out.
But it's the exact same nanoscale roughness in super hydrophobic and super hydrophilic. And so how do you represent the difference? Or just explain, why does the exact same nanorough surface act super hydrophilic in some cases and super hydrophobic in other cases?
For example, if you're going to teach your sister or brother to make these surfaces, how do you tell him or her how to make it?
And they're six, and they say, why? Why? Why?
So think about the experiment and what you did, and that might help answer their questions. So you made both of those
Okay.
surfaces rough, but then what else?
I think that I would tell my sibling to make sure that oxygen doesn't get into the little cracks within the nano rough surface, because I know that we scraped one of the surfaces to make sure that that wasn't the case. But it's the oxygen that specifically makes it hydrophobic. The oxygen that gets trapped within the nano rough surface, I know, plays a role in making something hydrophobic. Oh, okay.
We have a question over there.
Just to add on to that question, I guess, so how does non-polar and polar substances play a role in the decision between hydrophobic
Well, polar substances are more hydrophobic, and then the opposite goes for hydrophilic, I think. It's the other way around? Okay. It's the other way around. I said I think, so.
How would you engineer that using that understanding? How do you now make the two same rough surface? Now how do you make them different?
So, something. Okay, so if they're both rough, then one should be polar because water is polar, and that would make a hydrophilic surface. I mean, super hydrophilic, and the other would be super hydrophobic if it was non-polar because water is polar. Okay.
So the artist for this one, will you please say if there's anything that you'd like to add to what they interpreted about your image? All right. Well, let's give Minji a nice round of applause for her drawing. And then let's bring the next group up.
Hi, my name is Peter. This is Yen, and this is Eileen. So, one of the most important factors in our everyday lives is energy, and there are many forms of energy. And one of the main abundant, reliable, and pollution-free energy sources is solar energy. So why aren't we using solar energy if it's pollutant-free, abundant, and reliable? We're still using fossil fuels and other methods of energy production, such as hydrofuel and wind energy. Well, solar energy is made out of silicon, which is a expensive material because it doesn't react to oxygen, water, and acid. And silicon is very fragile when it's rigid, and it's really difficult to fabricate. So in Tuesday's experiment, we worked with an alternative way of solar cells, which are dye-sensitized solar cells. Dye-sensitized solar cells are inexpensive, but are lower efficiencies, 50% less efficiency, and are less stable.
Okay, so look at this picture. So this picture is a gumball machine. Sorry, how do I? Okay. So as we can see, this machine is approximately divided into 3 parts, and this part, there is a box that provides electrons to, gumballs to the machine. Sorry, spoiler. And and this part is the this part provides the gumball to the next state, and there is a platform, and the gumball lands on the platform, and the platform rises to a higher state. So the gumballs then transfer to this column, and it cannot be seen very clear, but I assume that there are a lot of tubes, so the gumball pass through the tubes to the this outlet over here, so it can be further delivered to the consumer. So how is this related to a solar cell? This is actually, this diagram presents the entire process of how a solar cell works. So first, this box here is the iodine. So in the experiment, we use iodine ion solution to provide the electrons to the circuit. And this part is the electrolyte. So the iodine replace the gumball, aka the electrons, to the electrolyte to replace the gumballs that have been sold out. So here, this column is a dye. So when the electrons reaches the dye, so the dye actually brings the energy state of the electrons from ground state to a higher state. So here the electrons actually have more energy. And this column, I assume the artist of this drawing is trying to represent the TiO2. So the TiO2 is actually where nanoscience comes in play to the solar cell. So I guess this long tubes represents the large surface area of these TiO2 particles, because as mentioned before, nanoparticles have extremely large surface area. And in this case, the large surface area is very helpful to the efficiency of the solar cell, is because the surface area actually can, so more dye can be applied to the surface area. So yes, so here there are 4 outlets of the gumballs, so I guess it means it is more efficient after the process of nanoparticles. And here we can see that there's a hand trying to put in a 10-cent coin into the machine, and here it says a quarter coin is not accepted. This is related to the wavelength, and the wavelength is closely related to the dye. And the dye is have different colors. So in our experiment, the dye was red. So it mainly absorbs the green light. So only 10-cent coin can be accepted.
Well, so there's two components that this author, I guess, didn't include, which was very important. This author didn't show voltage versus current. Voltage is how much energy is per electron, while current is the amount how many electrons are being collected. Well, and this author did really good in showing that it's a solar cell by showing it's a cycle, because the solar cell, the way it works is just like a cycle, and I think this illustrator did it really well. And the other thing that this illustrator did really well was showing the components of the dye and the iodine and this silver nitrate. However, just like I said before, this author, the illustrator, wasn't very clear didn't show the voltage and the current. So I think if the illustrator did that better, it would have been a really nice representation of a solar cell. Is there any questions?
We had to make it transparent so on the drawing. So what represents the sun on the drawing? The box represents the electrons that are being resupplied.
Here's the deal. I guess there are different interpretations about the coin, but at first we assumed that the coin was a sunlight, but then we realized that the artist wasn't trying to present sunlight as in coins, because there is a quarter coin that you cannot use, and there is no sunlight that you cannot use. So I guess the artist didn't include sunlight in this drawing. Then.
It interacts with the dye. Tell me anything about the dye. Well, the dye we used in the experiment was raspberry juice, so it absorbs all colors except red, and it reflects red back to our.
So now tell me something about what colors of light you can't use.
Well, white is an example of a color that you can't use because it doesn't absorb any. It doesn't.
So you just told me. So just tell me again. What's an example of colors you can't use? All right. What color didn't you use?
The raspberry juice reflected green light. Reflected green light.
Well, what did it look? What color did it look?
Oh.
To your eye.
Oh, red.
So what color did it reflect back to your eye?
Oh, red.
So what color didn't it use?
Green.
Red. So the dye we used in the experiment was red, so I guess we cannot use red light.
So 25 cents equals?
Red light? Right. So,
Oh, go ahead, sorry.
so like, there is no sunlight in the drawing, am I right?
There is sunlight.
Oh. I see, okay.
For time's sake, I think we'll thank this group and bring us the next. But as the next
next group is coming down, can the artist behind this one please say if anything was left out? So the bigger tube was the transparent conductor. Okay, cool, thank you. All right, next team.
Hi, I'm Sophia.
I'm Sia.
And I'm Melina. And our presentation was on photolithography.
So photolithography is a method through which someone uses light to basically create a certain pattern. So basically, photolithography is a top-down process. So it's basically going from a macro scale to a smaller scale. And so this process begins with a substrate, which is a polymer, and it's like the basis of this what we call a photoboard. And so then it's coated in a substance that is essentially like an active ingredient. It's called so it's any ingredient that you want to be part of the final product. So essentially, if you were creating a system of wires, you would want to use a conductive material such as copper. Next, this is covered in photoresist, which is basically a light reactive polymer. And on top of this, you place a mask, which is basically just the pattern that you want to have on the final product. Then this is exposed to UV rays. What happens is that whatever material was underneath the mask, it remains solid, basically. It's not exposed to UV light, which means its chemical substructure does not change. But whatever material was not underneath the mask, it's exposed to the UV light, and thus its chemical structures change, and it becomes soluble. So once this photoboard is removed from underneath the UV light, it's placed into a developer solution, which basically removes the photoresist that was on all of the exposed material. Then it's placed into an etching solution and stirred until all the excess material that is now soluble is removed. Once that is done and you have like a distinct pattern on your photoboard, there's a device called a stripper that basically removes all the remaining photoresist on the pattern. So, yeah.
So photolithography can be used to create integrated circuits or an integrated chip used in several of our technology that we use today, like computer chips or in your iPhone. Currently, there are, in the iPhone 7, there is 3.3 billion transistors, and transistors are composed, or each transistor is about 10 nanometers in size. Okay, so now let's explain this picture. So the first, in this diagram, in this diagram, we can see in the first quadrant, we have a person with a white piece of cloth, and we thought this represented the substrate, and they're dipping it into what looks like a cloth dye, and we thought this was depositing the photoresist, which we can see in the next picture. Then we thought the third picture was represented the mask. After you have the mask on the photoresist, cutting it out represents how we developed this pattern. Yeah, sorry. And then in the last, or not the last, the fifth picture, we see what we thought the person cuts it into a shirt, and we thought this was the etching portion of this process. And then next we have the person says it's maybe it's a little too dark, so they dump it in some water to rinse some of the dye away, and we thought this was the stripping process or removing the photoresist. sorry, not removing the photosynthesis, removing the photosynthesis, sorry. And then lastly, we see here, there's the final product, and it says here it's shorter and tighter. It's best running apparel with no, it gets cut off, but we think it says resistance. And yeah, so.
Okay. So we thought overall that this picture was really good and clear when showing this process, but there were a few concepts that they left out. One of the concepts is the idea of like using a clean room. So when working with nanoparticles and doing this experiment, we didn't use nanoparticles, but when you do this really small, you need to make sure that nothing disrupts or like gets into the design and messes it up. So you use clean rooms, and we actually visited a clean room, and in the clean room there were only like 100 nanoparticles per square foot. So a way to incorporate this into the picture would be to like show what not to do, and maybe have like some coffee that you spill onto the design, which would mess it up, and we don't want to do that. Another concept that we didn't think was really expressed was exposing it to UV rays after applying the mask and the photoresist. And we said that maybe you could like dry the cloth and give it some time to soak in the dye. And we also said that it might be better if the illustrator were to differentiate between etching and developing, because there were two different things.
Okay, forget it.
Yeah.
You mentioned UV light at the very end. How does the wavelength of the light that you use impact your ability to scale the nanoscale?
His question was, how does the UV light, can you repeat that, sorry?
The wavelength of the light you're using
Mm-hmm.
impact your ability to scale the size.
Oh, okay. So,
So basically, the wavelength it's basically scaled to how small you can create this pattern. So if you were to use something of like larger wavelength, like visible light, or like on the other end of the spectrum, then it
wouldn't work as well.
Yeah, the image wouldn't be as small. So to create something that's nanoscale, you need UV, just because of that wavelength. You require that wavelength, or that range of wavelengths. But if you wanted to create a pattern that's even smaller, then you would have to use like X-ray and even gamma rays, which people don't use as dangerous,
Yeah.
but
Able.
you would have to use much smaller wavelengths.
Yeah.
So you talked about the clean room environment. So why does that matter? How big the particles are, how many or how few there are?
Okay, so the question was, why does the cleanliness of the clean room matter? I guess. So it's really important when you make like or you make this with like nanoparticles, that you don't add in other nanoparticles that aren't supposed to be there in the air. Because if you were to do this, it would just like completely mess up what you were trying to make.
yeah, it could ruin the pattern that you put on your chip. Like little hairs, or I don't know,
Like dust.
Which could affect the whole thing. Yeah.
Let's see, topic. If that's a clean room, what, 100 nanoparticles per square foot? What's an unclean?
This right
basically,
here.
because there's.
So what are we talking about per square foot?
Oh.
Freak me out.
We actually asked that
Yeah.
question.
We asked one of our counselors.
Yeah, we asked one of
But
our
said-
counselors, but
His response was, that would take a lot of hard math, and right now I'm too tired. So.
Yeah, basically, yeah.
Oh, he said about 1,000,000 cubic feet. Is
Hundred.
a million nanoparticles per cubic foot. Is that how dirty? A dirty.
And, to be more technical, it's like these particles that are bigger than approximately a micron. Something like a pollen or a little smaller pollen, very small.
Okay, just to torment everybody. One of the key concepts of lithography is the idea of a mask. So how can you put that idea into this cartoon?
Said
Yeah, we said that we said like drawing it up. Yeah. Okay, so we said that the mask was the person like drawing out the pattern that he was gonna cut. So.
but we thought.
Right.
Yeah.
Mm-hmm.
Modify it to make more of a real, to capture the essence of what is a mask.
It could possibly be like a stencil, or maybe like we were talking about incorporating the exposing to UV light as a part of this cartoon because it's kind of not clear or not even there. So maybe you could incorporate how like you want to have some kind of design on there and then use some kind of stencil to like incorporate that.
Just within sewing, what could the mask be?
Uh,
No. Like, could it be? No.
either.
Fabric? I don't know. Thread? I don't know.
Oh, you
Is it?
buy like pieces of paper to like put.
Oh, okay, yeah. To to check the size. Is it for the sizes or?
Yeah. Oh.
Is that what? Yeah, we don't
Right.
sew, but. Okay. Yeah, that could be enough.
Hopefully a short one. The illustrator also spent a good amount of time writing shorter and tighter as a description of benefits for this. Can you relate this illustration to what in reality this might mean and why the illustrator put some effort into talking about the benefit of shorter and tighter for this?
So the shorter and tighter would relate to the like a smaller resolution because it's nanoscale. So that's basically the that's what that means in terms of photolithography.
What about directly to the application of the technology? Like, let's say this is the technology you're talking about. An illustrator might be suggesting that the result of such technology is shorter and tighter. Can you perhaps link that to the real world of why people would use a technology like photolithography?
So I think part of it would also be like making things smaller. Like one of our instructors gave us a lecture on basically like I see the microchips and how they've been getting smaller over the years, and that's in large part due to the nanotechnology. And so I guess that would be like not only have they gotten smaller, but they can also incorporate they can hold much more information. So yeah.
Artists, for this one, would you like to stand up and add any comments to what they presented? So resistance was a good element that they picked up on despite the challenge of not being able to read the scan fully. So thank you. So before the next group begins, I want to make a quick note to the audience so they know that two of our groups actually made drawings pretty much on the spot right after they did this experiment, because they did this experiment today, and then right after the experiment, the two teams did drawings and then traded drawings. So these teams had the least amount of time to both make their drawings and analyze the other team's drawings. So I just want to give a special shout out to those two teams before they start. So this is one of them. So if you guys are ready, then go ahead and start.
Okay, sure. Good afternoon. How are y'all doing today? Hope the traffic wasn't too bad. Anyways, my name is Marta Gonzalez. I'll be providing an introduction. Jason, he will be analyzing the illustration, and Ethan right here is going to be providing a critique. And our topic is supercapacitors, which we learned like 3 hours ago. Not even, okay. So supercapacitors is an energy storage device that relies on an electrical double layer to store charges. It's kind of similar to what happens if you rub a balloon against your sweater and it creates a charge differential. It somewhat works on that property, but some of the applications of supercapacitors include its use in elevators, and you probably won't see this in Southern California, but some buses have supercapacitors on their surface, so every time it reaches a bus stop, it'll charge and go on and charge and so forth. And in comparison to batteries, supercapacitors have a very high power density, which means it could charge and discharge very quickly. One disadvantage, or not disadvantage, but one difference from batteries is that batteries have a larger energy density, which means it could save more power in comparison to supercapacitors. Now, supercapacitors sound difficult, but they're actually extremely difficult. Fortunately, we have a little analogy right here that Jason will hopefully make a lot more simple to understand.
So, thank you, Martin. So, as they say, a picture is worth 1,000 words, and this one is no other. So, as you can see, this cartoon is split into 3 parts, 3 panels, top to bottom, and from our analysis, we have concluded that each panel represents one particular device that is used to store energy: battery, capacitor, and supercapacitor. So the top panel, which you can see has a bus, is representative of the battery. I'll start by explaining what the picture actually means. So you can see that there is one bus on the left side of the panel, loading up a whole bunch of people, and then ferrying them over to the destination. However, it takes them a full hour to do so. This is similar to the battery, and that the battery is able to store a great deal of electricity, a great deal of electrons inside of the battery. However, it takes a long time to charge, load the people onto the bus, and also a long time to discharge, get the people off the bus. Now, the middle panel is an analogy for the regular capacitor. As you can see here on the left side, there's a smaller group of people loading onto a single van. However, that van is able to get those people to the destination in half the time, 30 minutes. Similarly, capacitors are able to charge and discharge much faster than batteries. However, their maximum capacity for storing energy, their energy density, in other words, is lower than batteries. And then the bottom panel represents a supercapacitor. So as you can see, it kind of looks like they're on a highway with multiple cars, and those cars are able to get to the destination in, again, half again the time, only 15 minutes. And it also says first place, destination first place over here. So how does this relate to supercapacitors? Well, supercapacitors are able to store or just charge and discharge even quicker than capacitors. Now, this is where nanoscience comes into play. The electrodes inside of supercapacitors are blessed with a much higher surface area to volume ratio due to using nano-sized materials. As an object gets smaller, the ratio of its surface area to its total volume increases. And at the nanoscale, well, as represented by this picture, there are a whole bunch of cars compared to only one bus and one van in the other 2 diagrams, pictures. But here, there is a much larger surface area or spaces in order to store the electrons or people. So thus, the supercapacitor is able to charge and take in far more electrons or far more charge than either the battery or the capacitor are able to do.
So as Jason said, the batteries take a longer time to charge and discharge their electrons, and that's because to store energy, the batteries have to go through a chemical reaction, unlike the capacitors, which just require a physical reaction. And to show this difference, I think the artist or illustrator could have added maybe a pay station where they had to get onto the bus, just to show the difference between the longer time that the electrons have to travel to take. And on the bottom panel, you can see people standing outside the cars, but you don't really know what they're doing, unlike the top two panels where it shows arrows of indicating them going in and out of the cars. So I think they could have added arrows just to make it more clear. But overall, this analogy was pretty clear to understand and relatable. So yeah, thank you. Do you have any questions?
The supercapacitor, the electrodes that are inside the supercapacitor are usually made of materials that have, well, nanomaterials. And nanomaterials are, for example, one we used earlier today was called activated carbon. It was carbon that had been treated so it had many numerous fissures or fractures inside of it, which would increase the amount of total surface area that is exposed to whatever the activated carbon is exposed to. So these nanomaterials will just have much more surface area on the outside of their molecules or the outside of their particles, which enables them to interact in a much greater scale than normal materials would. So that's what makes supercapacitors much more have much more power density than capacitors.
Just to add on from the previous question, a general thing in nanoscience is increased surface area. Almost every question is surface area, including here. So with nanomaterials, you have a much larger surface area, and that gives a lot more opportunities for the charges to accumulate within the supercapacitor, and that's what makes the difference between capacitor and supercapacitor. One is made up of nanomaterials that enhance the surface area and allows the charges to accumulate much more efficiently and better than a capacitor.
What can you guys tell me about the speed of a supercapacitor versus a capacitor? Because I think there's some stuff maybe in the drawing or what you presented that was not entirely right. So which is faster, a capacitor or a supercapacitor? So you guys try first.
As a matter of fact, I think.
You've only got two choices, so you're 50% on to being right no matter what.
As a matter of fact, I believe the capacitor is technically faster at discharging and charging.
Yep,
Although
for sure faster, way faster. Like a couple orders of magnitude faster. So what's good about the supercapacitor?
the supercapacitor is able to charge up a significantly larger amount of energy.
Stores more. Little slower, still fast, stores more. Okay, so now can you tell me how you might modify the picture to capture those ideas?
Perhaps I could modify that 30-minute timer on the van picture to be 5 minutes, maybe 1 minute. So, just in order to show that the van makes it to the destination much faster than the bottom one, I guess.
And that would be pretty easy because what do you see at the bottom that might actually justify that?
It could be possible traffic. This whole.
Sure,
yeah.
Which you talked about at the beginning of your talk, incidentally.
So yeah, with a very small change in the labels, one could really capture all of the physics within this cartoon.
All right, let's thank our group. Next group, come on down. Artists, anything to add to what they were saying?
No, I think they covered it all.
All right, well, nicely done.
Hello, my name is Olivia.
I'm Jacqueline. And I'm Emily.
And we are circling back around to biotoxicity. So you've probably already heard a little bit about it, but biotoxicity is essentially the buildup of toxins in an organic body, and we analyzed how silver affects yeast as a biotoxin. We used yeast as our model organism, and yeast, one cool thing about yeast is it has a cellular respiration process where it takes in glucose and oxygen, and it emits carbon dioxide and water. So for our experiment, we used silver to kill yeast cells. We used 3 different forms of silver. And we measured their effectiveness by how much CO2 output there was using a manometer or a gas measurer. So we used 3 different types of silver. We used nanoparticles, silver powder, and Ag+ ions, which we took from silver nitrate solution. So the powder was too big, so it didn't kill the yeast cells. The Ag+ ions could effectively pass through the ion transports in the membrane, but it was a really slow process since the silver ions are positively charged. And then when we did the nanoparticles, we found that they were around the same size as glucose molecules. So what happened was the yeast cells were kind of tricked into thinking that these nanoparticles were glucose. So the the yeast cells kind of wanted these nanoparticles to come in, and what happened was the Ag reacted with the lipids in the cell membrane and broke down the membrane, effectively killing the cell. So that was the most effective in killing the yeast.
All right. So in the drawing, you can see 3 different animals, and their objection is to try to enter the building and kill all the humans in the building. So the giraffe kind of had some trouble getting into the building because he's too tall to enter through the door. And then the snails, they were able to get into the building, but they were too slow to catch up with the humans effectively enough to kill them all, because some humans were able to get away. And then in the final illustration, you can see that there are bunnies who seem very nice and sweet and cute. And so they're able to get in because they're able to disguise themselves as cute little bunnies. They're bunnies, but they're not cute. So when they get into the building, they turn evil and kill all the humans.
All right, so I'm just going to elaborate on that. I'm going to show you how the drawing is a really good representation of the concepts. So first, I'm going to talk about the gate, which says entrance and the humans inside. So the gate represents, the entrance right here represents the cell membrane, and inside are kind of like the yeast cells that we're trying to kill in our lab. And then all 3 different kind of animals with the 3 different kinds of silver that we used. So going one by one, the giraffe right over here was a representation of the silver powder. The snails were a representation of the silver nitrate, and the bunnies were the silver nanoparticles. So as Jacqueline said before, the giraffe had problems getting in because it was too big. And this is a really good representation because the silver powder, although it also can break off in ions, but because the powder is too big, it can't penetrate the cell membrane. So going to the snails, as you can see, the snails are able to get in and kill the humans, but as you can see, the snails are really slow animals. So although they work, they're very slow and not as effective as, let's say, bunnies. And I thought it was really clever that the illustrator had the snails saying words that are like, oh, be positive, we can do it. So they're saying really positive things to represent how the ion had a positive charge. So I thought that was a really clever way to represent that. And then in our last picture right here, the bunnies, they represent what Jacqueline was saying. It's they look super sweet until they get inside, and then they look kind of demonic and kill the humans. So it's kind of like a Trojan horse kind of thing, where it looks like, oh, it's a gift from the outside, but once it enters the actual cell, it becomes the downfall. So I also want to talk about how the artist did a really good job showing how using the animal size, although the snail is smaller than the bunny does a better job getting inside because it doesn't have a charge. That's why it's not saying anything, and the snails are saying positive things. So does anyone have any questions?
Glucose is much smaller. But the size of your nanoparticles and the size of food
In this case, like, because it's a lot slower because the ions have to go through, if they're going to penetrate the cell membrane, they have to go through the ion transport channels. So it's a little bit slower, so it's not as effective. So the silver nanoparticles are able to kill the cells faster and it's more effective.
But if you have the same effect at the end, if all the people are killed at the end, given the illustration, is faster always better? I'm not convinced. Maybe it's not your job to convince me.
Well, I mean,
There's a good answer to this. You guys think about it.
I mean, we were really looking at effectiveness in killing the yeast cells here. Like, yeah, the snails kill the yeast, but if you have a set time, then the nanoparticles are going to be a lot faster in killing the yeast cells. There's
My hint to you is, while the giraffes and the snails and the rabbits are having a party, what are the yeast cells doing?
Yeah, as like if you allow them to grow because it's really slow, even more yeast cells will appear and it's gonna make like, let's say like the disease even worse or something. So time is kind of the essence here. Yeah. Thank you.
So why are the instructors teaching you how to kill cells?
So like one of the real life applications to this would be like we had a lecture about how this was used in medicine today. So let's say like tumor cells from cancer. It's super like right now the ways we have to kill them are really ineffective because they also kill the good cells in the process of doing that. So as we use like nanoscale, so, like nanoparticles to do this, we're able to target them a lot better. And yeah.
In this experiment, it's very size selective, and so in the application you talked about does have some targeting concept. Just without any of the targeting concept, what would be the size selection by itself as solution for real applications? So this one, the experiment you didn't depend on targeting, as tumor cell versus a normal cell. It just tells you to kill cells.
Yeah.
So they all kill cells and effectively or non-effectively. What would be an application such that this would be sufficient?
Where might you use this in a lower tech application than differentiating cancer and regular cells? Where might you actually use this?
Yeah, just
Antibacterial.
antibiotics. You could just use it in medication to kill like viruses in the body.
We also know that the cells are just cells, right? We are made of cells. Why would we want to eat something that would kill cells?
I mean, I would say even just in like antibacterial wipes, or where you're trying to clean something that's organic off of something that's non-organic.
To worry about killing the normal cells if you're just using like let's say wipes because the objective would be to kill like the bad things. But if it was used in medicines, like I think the in the lecture they told us how like normally these like nanoparticles they have like a target on them to make sure they kill like at the minimum amount of healthy cells as possible.
But so your parents don't worry. Is this silver nanoparticles you made harmful to you?
Yeah, at large doses it can be, but if you like consume a small amount.
You could drink them, but don't.
Yeah.
But you can. Why?
It would start.
Did you drink them?
It would start to kill off your cells.
You turned blue, but.
Oh. Okay.
Okay. So I guess to make sure that our parents are comfortable that we're teaching you guys how to kill things that would affect cells, what is the reason why, say, human cells are not affected as much as the cells that you want to kill, like yeast and germs and bacteria? What is the difference between human cells and these bad cells? Maybe it's too long. I think we have another one. Right? So
we do have one more. Okay, we're going to leave for the last group. Where's the last group who's doing this? Somehow you need to work in their
Wait, does your body have like vitamins that break down the silver particles so it's not as toxic?
Are they
Next BioTox group, prepare yourself. Artist of this drawing, anything that you would like to add? Okay, so there was a lipid concept there that maybe they did not get to pick up on. Interesting. Well, thank you. All right, so next group.
Hi. Yeah, good afternoon, as Martin already said to everybody. But so this is our group. It's myself, Ethan Martin, and Maren Summers and Alex Witt. And we'd just like to talk to you about hydrophobicity and philicity. So in this nanoscience camp that we did this summer, our first day we got here on Monday, well, we got here on Sunday, but on Monday, our first actual class day and lab, we worked on superhydrophobicity and philicity. So as the other group stated, that's when certain substances are attracted, or the water is attracted to those substances or repelled from those substances. So how we did this experiment on that first day is we took 4 copper plates and we sanded them down to remove the oxidation layer so that we had a clean slate to work with. And with that, we dipped it in silver nitrate to give it the nano roughness that we could build upon later in the experiment for, it to just actually work. That's like the building block, the nano roughness, that is. So we used 2 different chemicals. I don't remember the name, but I can tell you that one was nonpolar and the other was an alcohol, which is polar. As we know, it's an OH attached to a carbohydrate, excuse me. And so what we did is we soaked 2 of the different plates, those copper plates with the silver nitrate. We soaked 2 of those in the 2 different substances. And then we had 2 other ones that one we put hand sanitizer on and the other we left just plain. We, of course, rubbed it down with sandpaper and then left it as it was. And we put a water droplet on every single one of those 4 plates. In doing that, we saw that the one with the alcohol was super hydrophilic because it attracted the water, as the 2 were both polar, the water and the alcohol. So what that showed us is that, it had a greater angle than 150 degrees, the contact angle, and that showed us the superhydrophilicity, excuse me, I mixed up the numbers. So that it was lower than 10 contact angle, and that's what showed us the superhydrophilicity of the alcohol. And then what we also did is we had the other substance, which was nonpolar, and we will put that on there. It formed the water droplet that was, of course, greater than 150 for the degrees for the contact angle. And the other 2, when we dropped it, were relatively close in contact angle, with one spreading out a little bit more and the other remaining more as a half dome, rather than the super hydrophobic, which was more of a perfect sphere, and the super hydrophilic, which laid as a puddle, as you'd see in the grounds.
In this picture, you see a boy sledding. So in the top left, you see him going down a steeper slope with a rough surface, and he's higher off the surface, which is indicating how the water was repelled. And then in the top right, the slope is still steep, but it's more smooth, and he's still repelled from the surface. And in the bottom left, it's a little less of a slope, and he's more attached to the surface, which would convey how the water droplet spreads out. And then on the bottom right, the surface is almost flat, and it's a little rough, so he's more stuck to it.
So we start with the top left image. This shows super hydrophobic surface. So he's sledding down the hill, and the rough surface is repelling the guy sledding off the hill. So it's like the surface repelling water. The guy sledding represents like the water droplet on the surface. The next image to the right on the top represents just a hydrophobic surface. So it's not as rough, but it still repels the guy sledding. And then the bottom left image is a hydrophilic surface. So as you can see, the angle of the hill isn't as great as the hydrophobic surfaces. So he's actually traveling slower, as indicated by the dashing lines in the images before. He has none in this one. So you can see that he's traveling slower, so he's more attracted to the surface. And then the bottom right image, he is definitely not moving fast, as the hill's almost flat and it's very rough. So, I don't know if you can, so it comes up better there than we have the image here, but the angle is, for the top left, is greater than 150. So the angle of the hill represents the water droplet, the contact angle of the water droplet on the surface. And compared to the bottom right, you can see the angle is a lot less. So that shows it's hydrophilic. I don't know, do we go? All right, so I don't know, what did we talk about next?
So what the artists didn't, they kind of conveyed all the concepts that we were taught here, but one thing was that with their contact angles, it kind of looks like here they're more doing it with the surface that the water was placed on, since it's seems like the water is the buoy and the surface is the snow he's sledding on. And so it looks like it's more measuring the surface, when with the hydrophobicity and hydrophilicity, you're measuring the contact angle of the water, not the surface.
And just adding on, one thing that I actually just realized now is that in the slope, as you see in the top left-hand or in the top left corner and the bottom right, you can see the bumpy, and that's supposed to be representing the rough nanoparticles that would be on the superhydrophobic and superhydrophilic surfaces. I believe that's all. That's all. Thank you.
So we had the water droplet on a surface, and depending on how hydrophobic or hydrophilic it was, the droplet would be more spherical or flat. Spherical being hydrophobic and flat being hydrophilic. So we would take a close-up image of that and then measure it with a protractor. You can any other questions? No?
So the fancier $10,000 instrument that we use here is the same. It's not a camera.
It is the camera.
I mean, not a phone camera.
But it is just a camera.
It is still a camera. And more stable than their hands, for example. And we can actually capture things that are really fast. Often you can track, literally, you can see the low feet of water vibrating. But yeah, it's $10,000 versus their phone. And you can see you are well spending on your phone.
I'm also going to say it's a lot harder to measure the angle with a protractor on the phone, because the angle on the protractor is a lot bigger than the phone, so it's a lot harder to measure the angle. So.
Okay, so which is there's a rough surface on the bottom right and a rough surface on the top left. How would you differentiate, how could you modify the picture to differentiate those two rough surfaces? And also, the artist sort of played fast and loose with angles here. So there is something a little funny about the way the hill and the angle are related. So could you maybe explain that?
Well, with telling the difference between like the superhydrophobic and superhydrophilic surface, and at least in our experiment, the superhydrophobic surface was like we dipped the copper in a chemical, like the abbreviation for the chemical is DCT, and so the I can't remember. It was a nonpolar chemical, and so that repelled the water. And then on the bottom right, we used a polar chemical to dip the copper in, which made it superhydrophilic.
Any ideas on how you could change the picture to reflect that?
You could show different types of like kind of coverings on the nano rough surface, since that's what we kind of did to when we dipped the copper.
Or maybe on the bottom, like rocks sticking out that the sleds could interact with. Sledding in bad seasons. But you see the idea I'm getting at. Okay, so the other question is, if you look at the top right and the bottom left, the ski slope is actually a very similar slope. But the angles are very different.
So one comment that I have is maybe the artist could have picked a side for the slope to go down, because it kind of, looking at it now, it's kind of a little confusing seeing that one way, you know, you're going down the hill to the left, and the other way you're going downhill to the right. That might be confused with like supplementary angles.
They're mapping the ski slope onto a different aspect of the angle, so that a big angle of 110 and a small angle of 39 actually end up looking sort of similar in the picture, because they're not mapping the slope onto, in fact, the top two would be overhanging cliffs. The way that, if you're mapping them all onto this slope. But it's important to bring this out because in a cartoon, people will visually take everything that's there. And so if there's a complication like that, you can actually lead people astray by making it simple. And so the one challenge to always do in a graphical representation of science is to map everything the same way onto whatever analogy you make, so that it's really simple to pull out the meaning. And if you change it, people won't notice, and they'll just assume things are a different way than the science is trying to explain. All right, let's thank our group.
Next group, come down, and before I switch the slides, artists, would you like to stand up and add any comments? No, it was really good. It wasn't terrible. We chose it because it was a good drawing. But none of the drawings that are being shown today are perfect, by the way. It's like Dr. Tolbert was saying at the beginning. It is pretty much impossible to get every single concept from the lab onto one of these drawings. So we can think about ways to try to modify the drawings, but there would still be no way to get every single concept on there. So this was a good drawing. We actually really liked this one. So good job, artists. Thank you. And next group, if you guys are ready.
Good afternoon. My name is Leo.
I'm Matthew. And I'm Tim.
So we're going to be discussing solar cells, or photovoltaic cells, which essentially work by converting light in the form of photons directly into electrons, which then provide electricity through both chemical and physical reactions. So solar cells are an important step in working towards a more sustainable and renewable future. Their importance derives mainly from the fact that the sun itself is one of, if not the most potent energy sources within our reach. In fact, A single expanse of solar cells in a sunny state, such as Nevada, is capable of powering the entire nation with solar energy. So to maximize the power capacity of our solar cells, nanoscience plays a major role. So it does so by greatly increasing the surface area in dye-sensitized cells, the solar cells we worked with, therefore improving the cell's ability to absorb and manipulate light. So given the potentially devastating effects of our current carbon emission levels on the climate, the importance of research in this subject of solar cells is more important than ever, despite the fact that it's very costly. So in this cartoon, which depicts a representation of the solar cell cycle through a baking process, we see these 3 illustrations in the corners, which we'll kind of get to later, and then this main cycle, which represents the solar cell cycle. So we have 2 ovens, one set to 375 degrees and one set to 50 degrees. And we can see that the pies being baked in these ovens are very different. The one on your left, they fully rise and they satisfy their consumers, which then results in money, or electrons, which then provides room for more materials, and that kind of keeps the process being continuous. It keeps it going. In this one that is set to 50 degrees, the pies aren't baked very well, and we can see the people are very sad and don't give any money, so they're not really helping in the process.
All right, so I'll be getting more into the science of this cycle of the photovoltaic cell. So this is a different type of solar cell called a dye-sensitized solar cell, which is different than the common silicon solar cell. So basically, this type of cell starts out with energy from the sun coming down onto a dye. In our case, we use a dye called anthocyanin, which comes from raspberry juice, and it's a red dye. And so basically, this reflects all the red and absorbs all colors, mostly being green, and uses this energy from the light to charge different electrons to bring them up to the next energy level. These electrons, in turn, are able to do 2 different things. One, go down an energy level, which is the easier thing to do, but it's also the thing you don't want it to do to provide energy. Or it can go on to the next step, which is to transfer onto the titanium nanoparticles. So the titanium dioxide used in the next step is so that the electrons that went up in energy level in the dye do not go back down. So also, why are these titanium nanoparticles instead of just a flat sheet of titanium? So this is so that since we know that nanoparticles have a much higher surface area, exponentially higher. So this makes it much easier to put more dye on it and thus absorb more light to power up more electrons to get a higher efficiency rate for the cell. After going on to the titanium nanoparticles, it goes on to another conductor. In our case, we use something called fluorine-doped tin oxide. And this basically is a conductor, but it has a special property where it's clear. So this has to be a clear conductor because it has to allow light to go through in order for the photons of the light to hit the dye, which then starts the process. After the FTO, or the fluorine-doped tin oxide, it moves along a wire, and this is where you'd have your power output, which can be a multimeter or a fan or anything else you want to attach to the solar power, and then to another FTO plate. From this plate, it goes on to an electrolyte, which we use iodine, and this basically restores the electrons back into the first dye and finishes that process.
All right, so I'll explain the analogy. We'll start with the middle cycle because that's the most important part, and then we can go to the corners. Now, starting from the beginning, our ovens. We have some ovens here delivering some lovely thermal radiation to our pies. You can see, Leo touched on this earlier, but the one on the left is at a much higher temperature than the one on the right. And so the pies are actually rising here, and they're not doing nothing here. So this is analogous to the fact that sunlight from the sun comes in a spectrum. There's a whole bunch of colors. There's a lot of reds, yellows, there's a smaller amount of blues and purples. And so what that means is the dyes that we use, they take a certain wavelength for the electrons to get excited. We use some red dye, the anthocyanin, and so those will, obviously, they reflect the red light out. That's why we see them as red. And they absorb the green the best. And so in that case, they take that specific wavelength for those electrons to be excited. But if the wavelength is wrong, or in this case, the analogy is the temperature, then the pies don't rise and the electrons don't get excited, and then they don't get transferred to happy customers. So going back to our correctly risen pies, they go down towards the happy customers. And in this case, the customers are like the titanium nanoparticles. They accept the pies, and then they keep it, and then they transfer it over as money. And this is analogous to sending the electrons into a current, which then they can get harvested into a generator to charge your battery or your Tesla car. And so after that, the electrons go back into the electrolyte, which is basically ingredients, right? This electrolyte provides the supplies for the next cycle to occur. And so this process goes on, and then electricity is generated. Now, up in this top left area, we have the person at the top that has 4 smaller pies, and he's pretty happy. And there's this person on the bottom here, he's unhappy, and he has just this one massive pie. And I think what that's trying to convey is the concept of nanoparticles and surface area. Now, the smaller pies, as you can see, they have a much larger surface area for a given volume. The amount of pie can be the same between the 4 pies, the 4 smaller pies, and the 1 big pie, but they have different surface areas. And so what that means is the smaller pies, they bake quicker and easier. And then so what that means is then the electrons can get excited more easily. And that's one of the reasons why we use nanoparticles. That's why we use titanium dioxide nanoparticles. Down on this bottom right, I think this is an example of voltage. And so what that means is you can see this top guy has a really big pie, and this bottom guy has just a really flat pie. And so in like the same batch, this person is making a whole lot more pie than this person. And then, so basically what voltage is that each electron has a specific amount of energy. Higher voltage means that that electron has more energy. And so what that means right here is if you're producing the same amount of pies, but you have a higher voltage, then you can generate a lot more current. Or in this case, deliver a lot more pies to a specific amount of people.
Thank you. Any questions? Okay, so his question was how to make these solar cells more efficient, I guess, and how to make them commercially available. So the main problem with dye-resistant sensitized solar cells rather than silicon solar cells is the efficiency. It's around 15% less efficient. And so the main reason for this is for actually 2 reasons. One, silicon has a much better, it's called a band gap. And so basically the silicon band gap is 1.1 electron volts, and the ideal band gap to absorb the most energy from the light is 1.2 to 1.4 electron volts. So silicon's really close to this and probably the material that's closest to this ideal band gap. However, the dye is not. And so another problem with this is how I mentioned before when you first start the process, and right when the dye absorbs the first amount of energy from the light, and it goes up in energy level, there's 2 things that this electron can do. It can either go down in energy level, which is what the electron naturally wants to do, or it can continue the cycle and go to the titanium dioxide nanoparticles. And so basically most of the electrons do go down that energy level and don't go onto the nanoparticles, and only some of them, which is the 15% efficiency, so only 15% of those electrons actually go onto the nanoparticles, and this leads to a major loss in efficiency.
I'll go a little more into like the comparison between this dye-sensitized solar cell technology and the silicon solar cell technology, which we see all over the place. Now, the silicon technology, it's more expensive to actually process the silicon, but as Matthew mentioned before, it's a lot more efficient due to that band gap being 1.1 electron volts, so it's very close to 1.2. It's silicon cells are really rigid, and so they're not flexible at all. But these dye-sensitized solar cells can be flexible, and so that might be one advantage as to why you might use the dye solar cells as to the silicon solar cells. One downside of the dye-sensitized solar cells is that, well, first of all, they're less efficient. We talked about that before. And they also, they're prone to degrading with exposure to the atmosphere. So that's more of an engineering problem, but obviously it's gonna increase R&D development prices up if you're talking about actually rolling this sort of technology out on a wide scale.
I think so. The titanium nanoparticles that we use are around 250 nanometers per particle, and that's to maximize surface area. And so I think it's more just of a material, it's harder to go smaller problem. And so more than just us not doing it. Because the smaller you can go, the better. Because the smaller you go, the more surface area you have, and thus the more dye you can get onto it. And so basically, I think that we would like to go smaller, except it's more of a material limitation rather than us not doing it.
Okay, so that sounds quite good. So first, they are 250 nanometers. They are 250 another unit. No, littler. Angstroms. They're 25 nanometers. And I could make you 5 nanometer titanium right now, but it would be worse. And so now your question is, why would it be worse? And I will help you with this by saying that 15% efficiency, it's not that only 15% of the electrons get onto titanium. Actually, probably about 80 or 90% get onto the titanium. But then, what do they have to do after they get onto the titanium? What is their fate?
Okay, so obviously they have to enter, they have to hit the film, the transparent conductor for it to enter.
Right between there, what do they have to do?
They fluoresce if you don't trap them.
No, between after they get on the titania and they get to the transparent conductor, what do they have to do?
Get there.
How do they get there? Yes, they have to get there.
Transmit.
How do they do it?
They jump from nanoparticle to nanoparticle.
Exactly right. Why might it be bad if the nanoparticles were smaller?
It'd be too difficult for them to reach the next one.
Yep.
They'd be insulated.
Goes up. And then that's one place you lose a lot of your electrons. The resistance in that process. And then also some of them, after they go into the titania, still go back to the dimolecule. So that's the other place that you lose. So that was really good. Okay, so two comments and one question. How you got voltage out of a little man with too much coffee is really, I'm impressed. I want to hear what the author says the bottom right corner is supposed to be. Also, I'm afraid I really disagree that they're pies. I think they're muffins. We'll leave that because pies don't rise and muffins do. But so this question is, these are solar cells. So how is the sun in here? And what aspects of the sunlight are captured and what aspects of the sunlight are not?
Okay, so the sunlight would be the heat from the oven cooking the pies. And so basically there are two different ovens in there, and one of the ovens has a different wavelength than the other one, which you can see through how close together they are. And so one of them's good, one of them's not. And so basically this change in wavelength would be the red versus the other colors of each of the wavelengths.
So now the question is, what aspect of what the sunlight does is not captured? So remember we said no cartoon captures everything. And part of your job is to say what is captured and what isn't. This is not a critique of the drawing. This is just intrinsic. Okay, so how does the oven
exciting the electrons in the dye.
So how is excitation captured here?
Would it be the happiness of the people? So they're getting happier. So you can see the comparison from when it's not, the wavelength's not correct, or ideal, to when it is.
I mean, I don't know. I mean, it's a cartoon. It's pies and flour. But but a concept that is not so clear is the idea that the electron goes from a low energy to a high energy, where the last one that didn't capture some of the concepts that this did showed that sort of lifting gumballs up. So every one of them will include some concepts and leave some out. But it's just interesting to point out where there's concepts that are.
Well, the change from the basic materials to the actual formation of the pie kind of represents the excitation of the electrons from their relaxed state to a more excited state. But
I could buy that, although a cooked muffin seems sort of relaxed to me. But I'm not.
you got it.
Okay. All right. Thank you, guys.
Thank you.
Artists, would you like to make any comments? I guess particularly in regards to some of your corner illustrations. Top left is surface area. Okay. Bottom left is current.
Wait, the most important question. Oh, are they pies or muffins?
They are pies. It has been confirmed.
I guess the blueberry, because you see in the bottom left, that could be like a blueberry muffin. So it's really.
We'll call it a puffin, a pie muffin. Everyone wins. All right, thank you. All right, good job. All right, next group. Next team, are you ready? Well, while they are coming down, since we need to power through the rest of the presentations, if everyone wants to stand up and take a little stretch, if you guys are feeling a little exhausted sitting, then please take a nice stand while the next group comes down. I believe we have 3 more, including this one. So this one and then 2 more. All right, everyone nice and stretched out? All right, so let's everyone take a seat again, please. And let's give this next group our same full, wonderful attention. So you guys ready? All right.
Good afternoon. My name is Kevin Park, and this is Michael Tang, and this is Carlo Ravelli. And today, we're going to be discussing the topic of photolithography. And photolithography is basically a process used in microfabrication. So for our experiment we did 2 days ago, we basically had circuit boards and we put film with the desired circuits on it, and then this was exposed to UV light, which allows for the copper not being protected by the film to be exposed, and then this makes it possible. Oh yeah, we have 2 chemicals, and so one is used to develop the circuit, and this makes the copper that's exposed to the UV rays able to be removed, and then the second chemicals is the one we mixed the whole circuit in, and then this removed the copper entirely, and we had to do it for a certain amount of time, around 7 minutes for all the copper to go away. And once the copper is scraped off, the copper not exposed stays, and it reveals the copper circuit, and it's exactly the same as to the design on the film.
All right, so now let's look at the picture analogy for photolithography. So in the first section on the top left, you can see three layers, so that might just be some like ground and some dirt and maybe a bit of rocks as well. So that would be representative of spin coating. So when you're setting this up, you'd spin coat some silicon to make it a really flat disk, and as you can see, these layers are pretty even and pretty flat. The second picture on the top right, we can see some trees. So these could be representative of a mask. So masking the photoreceptive layer, and then the sun on the next picture in the order could be representing UV light. So the UV light would react with the top layer of the soil, and then the wind could be the developer solution, which removes this layer. Going onwards, the wind could also be the stripping solution, which just blows off the top layer completely. So you're left with that kind of U-shaped piece of dirt. And finally, you have kind of a diagram of like a river meandering, kind of like a cross-section. And this could be, he actually wrote high resistance, so the dirt could be the high resistance layer, and as we know, rivers always take the path of least resistance in a similar way to how electricity would also take that path. So yeah, and you can see a little transistor here in the bottom right. So yeah, that could be, yeah, I think that's just supposed to represent a transistor actually. So yeah, thank you.
So I thought this picture was pretty effective in showing the solubility of the photoresist when it's exposed to light, and showing how like the film shields like some parts of the metal, but the transistor though, like it looks like it was just like hastily put there. Like they could have showed the functions of it. Like if I were to change this, I would like change it to like, I don't know, bridges or something to show the flow of electrical current. Yeah, I think that would have made this drawing better. Yeah, that's.
Any questions? Thank you. Just over here, Sarah.
Could you guys talk about positive and negative photoresist as they relate to this drawing and to your experiment? Or maybe just start with what's positive and negative photoresist.
I'm just kind of going off a kind of guess with a positive photoresist. So leave the parts where the UV light is shone on, and the negative photoresist would kind of leave an inverse imprint after the developer solution washes everything away.
Okay, and so in this drawing, where's the mask?
So
Or what is the mask?
the trees are the trees and the kind of tree roots are the mask. And so I think this actually shows positive photoresist, because is that right?
Forget the words. What's the concept? So here, tell me about where the mask is and where the stuff that's gone.
Yeah.
And your experiment. Tell me about where the mask was and where the stuff that was gone.
Okay, so the trees are in the same position as the sediment that's kind of deposited. So it's actually the opposite to what we had in our experiment.
That's right. In your experiment, the part that where the mask was that
Yeah.
was protected from the sunlight is the part that remained.
Yeah.
In
Mhm.
this, the part that where the mask is what goes away. And
Mhm.
both times exist and are used commercially, but it is the opposite of the one that you did.
Okay, sweet. Any more questions? Yes. So this is all. Everything is actually happening on a nanoscale. So this is actually supposed to represent transistors inside a microchip. So so in real life, this would. I think it's like 10 nanometers. It's like around that size. So that would be the size of one transistor, and all this is kind of creating one transistor. I don't know the answer to that.
You can make a little tiny mask. What could you maybe tell me about making a little tiny mask?
Do you use light to make the mask?
Nope.
No.
Wouldn't it be like harder to like shape the copper, I guess? Like, but it would be easier if you like did it on the mask though, because you you're basically like, because it's not like you're like carving it, right?
Well, actually, when you make the mask, you are carving it, but not with your hands, usually with an electron beam writer. So, but the question is a really great one. If you can make the mask, why do you need to do this? Why don't you just make the transistor the same way you made the mask? So, given that we said you have to carve the mask with electrons, what might you think about that process?
Is it because the copper conducts electricity? No? Okay.
Does it seem like an easy process?
Think about how many you have to put into a chip that's in your cell phone. How many transistors in your chip? You know?
It's like 30 billion.
Yeah, how long does it take you to draw the memory?
Take very long.
Yeah, it would take longer to like shape each copper piece than to shape like each mask, because wouldn't it be like easier to, because the mask is like much easier to make, right? So you could like produce more of it, I guess.
Harder to make. So the mask is really slow to make and really hard to make. And what does hard equate to in this world?
Expensive.
Expensive. So the mask is really expensive to make. So now why would you want to do this process? You make one expensive mask
Oh, because you can reuse the mask multiple times.
millions of times. You make one expensive mask, and then you can just shine light through it. One chip, shine light through it. Next iPhone, shine light through it.
Yeah.
So none of us would have a phone if you had to write every circuit with an e-beam writer. That would be way too expensive.
Wait, one question over here.
Since we talked about the mask and also how to manufacture a copper, so the question is, what kind of material used to make the mask? Is it copper or some other material?
I think. Do you remember what material it was made of? Do you know?
Well, you didn't use a real mask, so what was your mask?
I think it was a plastic of some sort.
Laser printer on transparency. Okay. What's a real mask made out of? Any ideas?
Light sensitive material.
Metal.
Metal the whole thing.
No, typically not copper. Then people use gold. I think people use platinum. I think the key is to be able to precisely, so the edge has to be sharp, and some metals are like copper. Not good.
Yeah, oxidation can actually make the edges uneven, so they use really expensive metals on top of that. A lot of them are chromium because they light easily. They're easy to ablate with the e-beam.
Yeah, so that's not a
There's a huge amount of consideration and restrictions, as you already pointed out. So that's why the math is so expensive. You're going to repeat the multipliers, you want that one to be 1 meter, 100% perfect in terms of precision.
But their math were just printed transparent.
As the next group is coming down, artist, would you like to add anything to what was discussed? Okay, cool.
So you were trying to have the trees protect the dirt
Oh.
from the wind?
Uh-oh. Was that just a screensaver? Maybe my battery?
This is not the plug. It's a USB-C. Technical difficulties. Please stand by. Yeah. Yeah, that's probably a good idea. Yeah. So while this is going on, while we figure this out, hi, I'm David Rich. These are my partners. We have David Schwartz and Akira Suzuki. And our topic today is supercapacitors. You've already been explained by another group, but we're doing that as well. So we actually made those this morning. As part of our lab, we created sort of a crude supercapacitor that actually worked. We were able to charge it using a USB, and then after turning the initial battery source off, it was still able to power a small LED without any external power. So we actually made our drawing is up there, and I believe Akira will explain that later. One thing you've heard a lot about sort of nano roughness and how surface area makes a big deal in these sort of things and why nano is so good. One thing we learned actually is that we had a very small amount of activated carbon, which we used for our supercapacitors, and 1 gram of the substance, which is almost nothing, has the same surface area as a football field, which is pretty nuts. Also, to give you an idea of nanoscale, the things that they're working with are about 1 to a couple hundred nanometers, and 1 tennis ball has the diameter of about 100 million nanometers. So these are things just on an incredibly small scale. To add to their examples of trains which can be powered by supercapacitors, another example that I found was a drill. So typically a cordless battery drill you can use it without a cord and it'll power, but a supercapacitor powered one will only last for half the time. However, it can reach its full charge in only 90 seconds. So that's another use of supercapacitors in normal life. They're very useful, and Akira can explain to you how they work.
Should I explain about batteries and like other stuff first, or should I just go right into it? Okay. Well, there's in this analogy of a pastry place. They have a jelly donut on the left, powdered sugar donut, and then a powdered sugar funnel cake on the right. And basically, batteries are slow, but they can carry more capacity and electricity. And so that's why the jelly donut was used. It holds more sugar. Well, sugar is like the symbolism of electrons in this analogy. But so jelly donuts hold more, but like it takes longer to pump. I think that was like the meaning they were trying to convey here. So then, on the other hand, all the powdered sugar stuff are much easier to like pour on the sweets. And then it says in the middle, I think, price reflects ease of sugar addiction. Addition, I'm sorry. Yeah. All right, well, yeah, so that the price is basically showing the difficulty of adding in the sugar into the sweets. So basically, supercapacitors use a lot of small scale particles in order to maximize surface area. So that's why, well, that's what we were told. So that's why they discharge and charge faster than the normal capacitor. That was our understanding. No? Oh yeah, they fixed it, didn't they?
They discharge faster than a battery.
Yeah, so basically it tries to use the same mechanics as a capacitor, which is supposed to discharge fast and charge fast, but have a little more capacity like a battery. I think that's what they were trying to convey, but yeah.
So one thing that was not displayed in this picture, which they could have put in, well, admittedly it would be pretty hard to include this, was specifically how a capacitor is different than a battery. So the difference comes from like the fundamental design of the battery versus the fundamental design of the capacitor. So in a battery, you have your electrons coming from a chemical oxidation reduction reaction, which occurs inside the battery, and this can take a comparatively longer amount of time. So in that time, you would get less electrons, which means you get less current, and then less power in a given time frame. So the capacitor doesn't do that chemical reaction. Instead, it has the ions in a solution. We used citric acid solution, and that allowed the electrons for the current to be conducted much more rapidly, and so that's how capacitors can provide much more power than a battery. So
I'd like to add one more thing, as well as the drill. Another application of supercapacitors, they can be used to discharge power very quickly. So they could be used in an electric car to give very rapid acceleration or something, because they're able to give out a lot more power in a much shorter time. Also, I'd like to give a quick shout out to our leaders back there, Patrick and KJ, and Tori should be somewhere around there, I'm not really sure, but they helped us and dealt with us this whole week, so thank you to them. Any questions? Sarah.
Okay,
Sarah.
Two questions for you.
Yes.
First, in light of the discussions that you had the first time, how would you change the prices on your food to reflect the ease of sugar addition?
Yes.
And the second one is, the group who drew this did something incredibly cool and insightful that you did not comment on, relating to the nature of the jelly donut and the powder and the funnel cake, and all
All right. Well, the first question was about the pricing of these things and the symbolism for that. Well, on the right side, there's a powdered sugar donut and then the funnel cake. They should be switched in price because the funnel cake is supposed to be symbolizing a supercapacitor because of its surface area. But in the previous discussion, we said that a supercapacitor is actually slower to discharge and charge than a normal capacitor. So, so the $2 and $1.50 should, yeah.
I guess jelly would be quite a sugar-dense way. Also, it is put inside. It is not outside. So if you basically the way these supercapacitors work is rather than storing the energy in chemical reactions. Yeah, I know, but so the supercapacitors actually just store the electron on the outside, on the surface. That's why surface area is so important, because if you have a lot of surface area, you'll have a lot of space to store electrons. So if the powdered sugar in this metaphor are the electrons, then they would be stored on the outside of the funnel cake and the donut, where the jelly would be the same as a battery, which would be some sort of like fluid inside of a battery.
The battery does a reaction inside the material of the battery. All the capacitors are a circuit storage technique. So I just want to flag this as an example that anybody could understand. Like if you're trying to convey to a 6-year-old, how is a capacitor different from a battery? You can say, it's like a jelly donut. The battery happens on the inside. And the capacitor is only on the surface. That's an analogy that anybody can understand. So.
Yes. Any more questions? Oh, one more?
I don't know how much you guys talked about commercial applications, but a car has a battery.
Sure.
Which is a deep cycle battery. It can start a car many times before it dies. But you can also buy things that just jumpstart the car if your battery is dead. Are capacitors more likely to be used in those? I mean, are they used commercially like that?
I am not sure if they're actually used in those products specifically. I know that they cannot be used in, well, electric car batteries at least, because they don't have the capacity for long range delivery. I'm not quite sure whether like immediate jump starters of batteries would be used with supercapacitors. However, I think that they don't fully recharge the battery. They just give it enough to start a motor, and then the alternator will charge the battery on its own in the car. So I don't know if you guys know any more about.
Wait, were you referring to using one car to start another car?
No, he was referring to like an external source. Yeah. Yeah.
Yeah, the jump starter is a little bit better because it's still cheap enough for you to buy. But I think in terms of the application for the jump start, it mostly to get enough voltage to break down and cause that to go. And so that doesn't leverage much. You're just trying to create enough of a voltage to create that one spark to go. And that's not the best use of a supercapacitor or anything, because you can generate enough voltage, whatever circuit you make, enough current to be able to generate the voltage. But in terms of maybe the other how to apply supercapacitor now, I think all of you guys have a supercapacitor in your pocket, so maybe you can just explain that quick application, and then you can probably quickly see how people are using supercapacitor. So you can show one demonstration of your supercapacitor you might have.
You mean the phone?
I'm gonna infer that you meant a phone by that.
So
yeah,
it can charge quickly? So one benefit, one. Is
The
it
charging dock? The charging dock? Oh. Inside the phone. Yes.
The flash?
Oh. I thought smart.
Well, the flash is like an instantaneous like flash of light. So like I mean, you don't really need the capacity to like shine like extensive amounts of light. Just flash in there. That's
Like
it.
a large release of energy at one moment.
Yeah.
I thought you were just taking a picture of us. Actually, I didn't get the metaphor.
I think in the docks, right, they have these elevators that actually uses a lot of energy because they have to lift something heavy so high, and that's a lot of energy. And then you try to capture it when you drop it down because that's free gravity energy, except that there's not enough material to be able to capture that energy fast. And so normal battery won't be able to capture that, recover that energy, but something that can fast and quickly take those powers, either deliver or capture, and be able to do that. And so these are all the applications that are coming out right now. Coming out. But Tolbert and her colleagues are experts in this in the world. I mean, all electricity square paragraph. So she can probably speak more about this than I would.
Oh, we can't, because we have to go on, because we're almost out of time. Yes.
Thank you very much. Thank you all for coming.
All right, as our last group comes down, artists of this drawing, anything you would like to add to the conversation? Or did they pretty much get everything? They got it all? Good analysis. All right, nice. All right, so last but certainly not least.
Hey, hi. I'm Naya. This is Ben, and this is Eric, and we're going to be talking for the third time about the biotoxicity of silver nanoparticles.
So what is biotoxicity? Biotoxicity is basically what the name implies. It is it's like how toxic a material is that can affect life, or like in this case, cells. And then, so why are we using silver? Silver has natural antimicrobial properties, meaning that it has the ability to kill microorganisms such as bacteria and viruses and fungi. And the utilization of these health benefits can be seen throughout history, from the ancient Romans, who introduced silver into their water supply to keep the water supply fresh, to Chinese emperors who ate with silver chopsticks, and then to silver leaves that were used in World War I to treat wounds, and then up to today in modern medicine. But how does biotoxicity relate to nanoscience? So biotoxicity, the effect of silver varies greatly depending on the size of the silver that you use. So to test this, we used 3 different sizes of silver. We first used a large powder, a relatively large powder. We then used a nanoparticle, which was on the nanoscale, and then finally we used an ion, which is much smaller than the powder. The large powder. The nanoparticle has a much larger surface area than the powder because it is so small. Larger surface area to volume ratio because it is so small, and the importance of that will be explained later. And then the ion, although even smaller than nanoparticle and therefore having an even larger surface area to volume ratio, expresses its own challenges in how small it is.
Okay, so just an overview of how silver is toxic to biological cells. So depending on the size of the silver, it can penetrate through endocytosis or membrane protein channels. And so once inside the cell, the silver ions take an electron from oxygen, which catalyzes formation of a reactive oxygen species. And so this reacts with the lipids in the phospholipid bilayer in the membrane of the cell and ruptures it, so it kills the cell.
So now for the fun stuff, we're going to be looking. Oh. Okay. So over here, you could see we're at a TSA check, and over here we got some bad guys who are going through security, which is very bad, and they're like the silver like nanoparticles, and you can see they pass through security. so they're supposed to, like, they're the silver nanoparticles. And once they get through, they kind of like, they take off their mask, which is like the cell thinking they're glucose, but not because they're the same size, because they're pretty similar in size. And then so they go onto the plane, and then they like blow it up, and which is bad. But over here, and that was like very efficient and easy because they had like no hassle through like security. But over here we got the silver ions, which is the bad guys. They're like going through security, and then kind of the ion channel. Instead of in this picture, the equivalent would be the special TSA room, where they, you know, suspect something's going on, and actually something is going on, but they still don't stop them. It just takes them a little longer, a little more time. Maybe a couple hours or so in the real world. And then, so like, they go onto the plane, and then they still blow up the plane. So that would just be like this, the silver ions in the ion channel. And then over here, we got someone who can't get on the plane because he's the silver powder, and he's too big to actually get into the cell, the yeast cell, and cause any damage. So then the plane is safe and fine, and the plane is not in harm's way. So, yeah.
Okay. So one thing the cartoon didn't address was how, in our experiment, we actually monitored the living cells to tell the effectiveness of the different sizes of silver. So we used yeast as our living cell, and so we measured its carbon dioxide emissions, since it is an aerobic microorganism and it goes through cellular respiration. And so the presence of carbon dioxide indicated like the live cell and the failure of the silver powder. So when we added silver powder to the yeast, the carbon dioxide there was carbon dioxide being produced because the silver powder was too large to penetrate the cell wall. And it did slow down because the silver powder still worked. It just took the longest and was the most inefficient because it had to go through a lot to break down into individual ions. And so when we added the nanoparticles, the carbon dioxide production halted since the cell mistook its size for food and absorbed it, so it effectively poisoned itself. And so when we added silver ions, these were the tiniest and were able to pass through the ion-gated channels, but this took a longer time. And so the carbon dioxide continued for a while, then stopped once the silver ions eventually penetrated the cell. Yeah, that's, thanks. Questions?
Any?
We can start with the question we told you.
Oh,
Which is,
yeah.
why are we not poisoning you all with stealth bombs?
Yeah.
During
So we,
Oh.
do you want me to do it? So I actually looked up, there was an experiment in 2010 by the Chinese Academy of Sciences, and so they exposed human cells to the silver nanoparticles, and only 4 to 9% of the exposed cells were ruptured. And so when we were learning about this, we learned that cells can cope with the silver nanoparticles, and they use vitamins A, C, and E, and the enzymes superoxide dismutase and pyridoxase. And we also thought that we could factor in how humans have an immune system and like specialized cells to target like intruders into the body, while like yeast cells do not.
I think also part of it is that our cells are not quite as stupid and don't eat everything that comes their way.
Yeah.
Where some microbes are just,
I-
that's only one microbe, they'll never die, they'll just fry.
I think like one part of it, yeah, just like what you said with the yeast cells, is everything was very optimal for them for a reaction. So like the water was warm, there was sugar and plenty of space for them to actually expand. But you know, in the body, it's not necessarily trying to constantly do that. And maybe one more thing is when we talk about nanoparticles and their surface area being way larger, we don't mean one small nanoparticle. We mean like if you fit a bunch of tennis balls in a basketball, that has a larger surface area because it's a lot of little particles. We don't mean one small particle has the same amount or more surface area than a larger object.
Your last chance, parents, one parental question to end. Last question of the whole time.
Any questions? Maybe? Over there? Yeah? Yeah, we had a lot of fun.
Yeah, we had so much fun.
Lots of fun.
What was your favorite experiment?
Favorite? I think this experiment. I think I would have to say that, but I would maybe not experiment. Maybe I think the coolest thing we've done is probably look at the electron microscopes. I think that's pretty awesome, and you know, they're really cool. You could look at really small things. Anything else? Sorry.
And before we end, artist, so we can recognize you and your work, and if you want to add anything, who's the artist behind this one? Yay! Did they miss anything, or did they capture your drawing? Any other concepts you had? So that was pretty much it. All right. Well, with that, thank you guys so much for all of your patience while we started a bit late because of some audio-visual things. And we have a lovely reception for you in the CNSI lobby. But prior to that, Dr. Tolbert wants to say something.
And the one thing is, the one person we haven't gotten to thank yet is Rita, who has been in charge of every aspect of this program. So, awesome job. Now you get to go to the reception. Back at CNSI.